In the previous video, we covered mitosis and the interphase. Now we're going to cover meiosis. So, what is meiosis? Similar to mitosis, it is a process that splits and divides cells to form daughter cells. However, unlike mitosis, it produces 4 daughter cells. Now, it's important to know that these 4 daughter cells are not identical, and they are haploid cells, otherwise known as gametes. Now, we've gotten the basic definition of meiosis out of the way, but how does it actually proceed? So, we're going to start focusing on the steps of meiosis. Now, I'm going to draw out how it proceeds just as a rough diagram. So you start off with a cell. All right. So this is the very basic outline. Now we're going to start drawing chromosomes. So in this stage, we're going to have some amount of chromosomes, and then in this stage, that's actually going to become double of that. So they replicate, and we have chromatid. So you get sister chromatids, so they replicate. Then we're going to go to the next stage where mitosis, pretty much mitosis, at least that's what it looks like, happens. So you get 2n, that, like that. And that. Now, at this stage, you're going to get some very odd looking cells which are not identical to the parent cell. So we're going to have that. Now, let's actually start labeling these with numbers. So you're going to get this as your 2n. This is going to become your 4n. This is going to go back to your 2n, but it's going to be slightly different because in here it's 4n, so it's 2n as in 4 individual chromosomes or a total of 2. So n is represented by a singular chromosome. Actually, technically half, like 2 of these individuals will form together to form 1. Then you're going to get this being n. So all of these, the bottom row are going to be n. This is 2n. This is 4n. Now, where do these steps occur? So to go from here to here, you have your interphase, where all the cell contents and stuff multiply and you get your fully grown ready cell that's about to split. In this phase, we're going to call this, it is called meiosis 1. Cis 1. You're going to have, this is actually quite a long set of complicated steps, which I'll go into later. And then finally, to go down to the second one, this is produced by meiosis 2. Cis 2. So this is pretty much your outline. So you start off, this is interphase. Meiosis 1 should actually be here, and meiosis 2 should be there. So that's what produces these cells. Now, how does this actually happen? Well, just take a second to write this down. notice what's happening, and we're gonna move on to meiosis 1, which is what we're focusing on. So in meiosis 1, you're going to have a couple chromosomes. So say this and this, as well as, oh, kind of an accident, okay, as well as this and this. Now they're going to multiply to give you this and this. Now once we're at this phase, this is done in the interphase and the synthesis phase, we're going to move on to the prophase. So in the prophase, we're going to have these sister chromatids. So these are sister chromatids. This and this are sister chromatids. For simplicity's sake, we're going to erase this. So we're going to have sister chromatids, and likewise these are sister chromatids. We're going to actually have them start to cross over. So we're going to have them start to look something like this. Now, once they've crossed over, these sections here, I'm going to circle, cross over, do in chiasmus. You don't have to know the name, but you just have to say they cross over to form bivalence. Now, once we formed our bivalence, we're actually gonna be in a position to begin our metaphase stage. So in metaphase, let's just move this over here, we're gonna have the same stages or the same steps that normally occur during mitotic metaphase, where the chromosomes, so let's say we have sets of crossed chromosomes now, that we gonna represent like this, and then like this. These crossed over bivalents begin to line up at the equator. So this is the equator, and they begin to line up. Now, from this point onwards, the steps that occur are pretty much the same as mitosis. So we're gonna erase this. Just take a second. We're gonna erase this, and we're gonna explain how they move from here. So within in mitosis itself, we're gonna go. We already had them line up at the equator. So now we're gonna actually have anaphase happening, where they start to split off into the equator. So you're gonna have these guys. They're gonna split and be pulled to the equator by microfibril tubules, which we already know what those are. Now, so pulled to equator, and this again happens by kinetochores, and it's important to remember that these are still crossed over. So you're gonna just remember that as crossed bivalence. So, all right. Now what happens? Well, you're gonna go into your next step, which is telophase, where you're gonna have cells with these different crossed over chromosomes. So we're gonna have this and this, and this. So now you're gonna have your two cells that have that, and cytokinesis occurs. Now, what's important to note is from here we go into, so this is finally, so I'm just gonna telophase, cytokinesis. Now, what happens here is you have two cells with these bivalent chromosomes that now require to change into halves. So they need to split so they can form haploids. Because at this present moment, the number of chromosomes inside of this is still going to be, and this is just a graphic representation, but the number of chromosomes is still actually going to be 46, right? So there's going to be 23 distinct pairs, but we don't want that. We actually just want 23 chromosomes, or something and a half pairs. So we're going to erase that. So there we go, 11 and a half pairs. We're going to erase that, and at this point let's just write it here. Still 46 chromosomes, 23 pairs. All right, now this is going to be your final steps of meiosis 1. Erase that and go on to meiosis 2. Now, in meiosis 2, something very interesting happens. The steps are relatively similar, but in prophase, the sister chromatids that condense are going to just be split up. So let me look for the wording. So the sister chromatids are going to like not multiply in this case. So we're not actually going to have, so just write this here. Interphase will not happen prior to meiosis 2, and that's significant because we're not going to actually have the duplication where you're going to get 46 going into what, like 98 or something. Yeah, no, 92. So you're not going to have the 46 going into 92 chromosomes so that they can split. Instead, you're going to have the 46 remaining as 46, but they're going to start dividing now. So 46, we're going to have two 46s from the previous step. We know that there's 46 and another 46. That's going to go into 23. That's going to go into 23. That's going to go 23. That's going to go 23. So you're going to get 4. But how does that actually happen? Well, let's erase this. Just remember this point. Interphase will not happen prior to meiosis 2. And let's actually draw it out. So prophase is relatively the same. The sister chromatids will condense. So we're going to have this looking like this and this. And then you're going to have another one. It's going to look like this and this. And this all stems from just what was happening during our last stage. So if you remember, there were two full cells. Now, from this point, we're going to actually go into something interesting. So metaphase happens, and this spindle fiber, the spindle fibers from the centrosomes will start to come out. So we're going to have this. Centrosomes go here, and in metaphase, the spindle fibers start to shoot out in both steps and pull apart at these at the kinetic goal. So you actually have just a green and just a blue. Sorry, it's wrong step. So this is the metaphase. So you're gonna have just the blue line up on this side, say, and the green lining up on this side. And then likewise, you're just gonna have the blue on this side and the green on this side. Now, from this point on, they split and you get to your next step, which is the anaphase. So in the anaphase, they're going to start actually being pulled to the poles. So you get your singular blue and your singular blue also here, as well as your singular green and your singular green, all being pulled finally by the centromeres to the centrosomes. Then what you're going to get left with is actually going to be the T, like the final part, where you get two cells like this, that have one green, one blue, and one blue and a green. So this is going to be your progression, and the same thing will happen here. So at the end, you'll be left with, let's just go here, dot, 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 dot. So you're just going to be left with four copies. Well, they're not going to be exactly copies because obviously they've crossed over and they'll have different patterns, but you're at the end going to be left with four haploid, which is, let's write this here actually. So four haploid cells, and these four haploid cells are actually just going to be 23 chromosomes each. So 23 chromosomes. And obviously when people copulate, or animals copulate, or even plants through sexual reproduction, when they merge with one another, so in this case humans, they're going to join, and the 23 from the egg, the 23 from the sperm would ideally make a total of 46. So that's going to be your zygote. This is your gamete, or one of your gametes. So this is basically just meiosis, and let me erase this and quickly write down the importance of meiosis. So what are actually the main reasons for meiosis? So genetic, take variants, so that not everyone is just the same, because otherwise, say you had a same plant population that wasn't reproducing sexually, and every plant had the exact same genetics, a singular disease could wipe them all out. And we look at that actually in ecosystems. And then we go here, we also have it as being two parents, which again, similar idea, similar concept. And where it happens is sperm, ova, but in a nutshell, sexual reproduction. Sexual reproduction. All right, this is just a brief overview of meiosis, and that's gonna be our third topic that we've covered within the cell cycle.